Hi, my name is Emily, and today I will be presenting a spectrum of antimicrobial activity of 3,4-dihydroquinazolone compounds. So bacteria is constantly mutating and developing resistance to the antibiotics created to inhibit their growth. A series of new 3,4-dihydroquinazolone compounds were recently synthesized in the Mosey laboratory. They were primarily screened for bacterial growth inhibition properties against S. aureus. And the objective of this study is to evaluate the 10 compounds showing the greatest S. aureus inhibition from the previous studies for their ability to inhibit growth of additional gram-positive and gram-negative compounds. This will determine if these leading compounds exhibit broad spectrum activity or if they are particular to specific bacterial strains. It will also aid toward a better understanding of the mechanism of bacterial growth inhibition by these 3,4-dihydroquinazolone compounds. So in this study, there were 4 bacteria observed. One was gram-positive, that was E. faecalis, and then the other 3 were gram-negative, that was E. coli, A. baumannii, and P. aeruginosa. So one of the major bacterial targets for antibiotics is the cell wall. The size of the cell wall can cause bacteria to have a stronger resistance to the antibiotics than others may have. So a gram-positive bacteria has a thicker multi-layered peptidoglycan layer, while a gram-negative has a thin single-layered peptidoglycan layer. So there was 10 compounds observed in this study. These had shown good inhibition against S. aureus from the previous study. This research was conducted by first creating compound serial dilutions. There was 10 dilutions total. The first well had 100 microliters of the compound. 50 microliters of that compound was then transferred into the next well, which contained 50 microliters of DMSO. And then it was just carried out until the 9th well, which then left the 10th well to be solely DMSO. Next, I had to create the bacterial suspension. All of the bacteria was streaked on a TSB plate and incubated overnight at 37 degrees Celsius. And then 50 milliliters of sterile Mueller-Hinton broth, or MHB, was inoculated. The concentration of the bacteria was determined by measuring the absorbance at 625 nanometers, and the wavelength reading had to be between 0.08 and 0.12 in order to move forward. And then 0.5 milliliters of that solution was then transferred into 100 milliliters of sterile MHB to create the final concentration of the bacterial suspension. So this is what the assays looked like. So 5 microliters of each of the dilutions were added into the corresponding wells on this plate. 195 microliters of sterile MHB was added into the bottom 3 rows to create the control. 195 microliters of the bacterial suspension previously made was added into the top 3 rows. And then around the sides of the plate, 200 microliters of sterile DI water was added into those cells. And this plate was incubated again in the 37-degree incubator overnight. This table presents the results of the broad spectrum analysis. The software GraphPad Prism was utilized to analyze the absorbance reading from each of the plates. That absorbance reading was taken and a graph was made of the bacterial inhibition. And from the graph, the IC50 values were able to be determined. This value reports how much substance is needed to inhibit a substance's ability to reduce biological growth by 50%. So there were 4 compounds showing decreased activity, suggesting growth inhibition. And those compounds were SD1033, SD1123, TW1169, and then TW1139. Three of those compounds were inhibited by E. faecalis, the only gram-positive bacteria, while one of them was inhibited by P. aeruginosa, which is a gram-negative bacteria. So SD1123 was the only compound showing complete inhibition, and this is because the graph reached the baseline. So the other three compounds have the greater than BL at the end, which means that it was above the baseline, stating that complete growth inhibition was not observed. And then all of the other ones that show a greater than 125 represents that there was an increase in potency. It was not inhibited by the compound whatsoever. And then the S. aureus data in the first column was previously determined. So these two graphs show SD1033 and TW1169 against E. faecalis. As I stated before, they did not reach the baseline, so there was not complete inhibition from these compounds. And then these two graphs show SD1123 against E. faecalis and TW1139 against P. aeruginosa, the gram-negative bacteria. SD1123 did reach the baseline, showing complete inhibition of this bacteria. And then TW1139 did not reach the baseline, showing some but not complete inhibition of the bacteria. So overall, the compounds did not show broad spectrum antimicrobial activity. E. faecalis was shown to be more susceptible to growth inhibition by these compounds. And TW1139, since it was inhibited partially by a gram-negative bacteria, it possibly illustrates a different target for the gram-negative bacteria by these compounds. Cell wall biosynthesis is a plausible target for these 3,4-dihydroquinazolone compounds, but further studies need to be conducted to limit potential targets within gram-negative bacteria. I would like to thank Selena Malcolm for her work with the 3,4-dihydroquinazolone compounds. The results of her research with S. aureus is the foundation of this study. I would also like to thank Dr. Mosey for advising me and Dr. Hutchins for helping me in the laboratory. Thank you.